Well, this is an exciting morning because I get to hear great grand rounds given by the two Jeffs, Jeff Square, Jeff Herron, PhD, and Jeffrey George Ojemann. I am not going to take up all their time talking about them, but I do have some things to say. So, first of all, Jeff Herron is an assistant professor in the Department of Neurological Surgery. He's also an adjunct professor in the Department of Electrical Engineering at the University of Washington. He did his bachelor's in science and electrical engineering at the University of British Columbia and then his PhD at the University of Washington. And he went off to be an executive at Medtronic, doing scientific discovery with them, and then got recruited back by Jeffrey George Ojemann, who has some heady titles. He is the chief physician executive of Seattle Children's for the last 2 years. He is the chief medical officer of Seattle Children's for the past 3 years. He sits in the George A. Ojemann Chair for Epilepsy Research, given by the University of Washington and the Board of Regents. He was past surgeon in chief at Children's Hospital for 3 years and past senior vice president. He's now, as I said, chief physician executive and senior vice president. He's still a neurological surgeon. He was for 10 years the division head of neurosurgery at Seattle Children's Hospital. He is the vice chair of neurological surgery, the head of the discovery, vice chair for discovery. I can go on and on about these two gentlemen, but I rather they talk about their brilliant work together. I so admire what they did. It was an exciting week last week when Andrew Coe and Ben Grannon implanted a deep brain, a device to help rehabilitate a patient after stroke. They're going to tell you the 50-year bench to bedside story. Thank you, Jeff and Jeff, for being here. I hope I didn't take too much of your time, but this is exciting, really exciting.
Yeah, thanks, Rich, and thanks for your enthusiasm. Can you hear me okay?
Perfectly. We
Great.
hear you and see your slides.
Okay, perfect. And this is, yeah, it is exciting, and we always leave time for your very, very generous introductions. So I'm with Jeff Herron. We figured out how we're distributing the slides late last night, so bear with us because we're both pretty enthusiastic about this. Jeff, who's recently became associate professor and also has appointments in electrical computer engineering, and has emerged as a national leader in getting these devices launched in creative and important ways. So thanks everybody for being here. And what I'm going to do is go through some of the history of operant conditioning, which truly is a half-century story or
Hey,
more
operant conditioning. Wow, you see.
at the University of Washington. This is lots of enthusiasm. And talk about how we've used clinical recordings to drive brain-computer interface, and then the regulatory journey to a clinical trial, which is in and of itself story-worthy. So we will accidentally fall into jargon, and I apologize ahead of time. This is my attempt to make sure that we're careful. A nice old drawing with new technology put together, which is one of the themes today. But when we talk about EEG, we know what that is. This cases that the residents now do are primarily stereo EEG with thin devices that go into the parenchyma. Corticography is still a mainstay of many recordings and was how we diagnosed epilepsy and treat some diseases with stimulation, and that's on the surface of the cortex. And then a lot of what you'll hear in the news and the scientific and lay scientific and hype scientific literature is around microelectrode arrays, which are attempting to record from field potentials around individual or small populations of cells, and that's what we're calling microelectrode arrays. And so these were used because they give you really, really strong signals. So these would be an example from electrocorticography. You can see very low amplitude, high frequency changes that are the onset of a seizure. You can record interictal, but and you can break these down into different bands. They're actually named for Greek letters in the order they were discovered. So the alpha rhythm recognized in both animals and humans at the turn of the previous century. being these 8 to 12 hertz rhythms that are very prominent in the back of the head, and so forth. We'll talk a lot about beta, which is roughly in this 12 to 24 hertz range. So when you have a full signal, which is at the top, you can break it down into these individual components based on their frequency. So we're looking at oscillations that occur roughly 12 to 20 times a second. And what's been known for quite some time is that these will change, the power of these signals will change with activity. So if you do an EEG and look at just the beta power, that 12 to 20 hertz or so, and move your hand on the opposite side, you'll see a decrease just before the movement, a decrease that's sustained throughout the movement in the power of beta over central cortex, and that rebounds as soon as you stop the movement. So actions can create a signal change that's detectable even from EEG. And so as we look at these different levels of recording and look at the clinical one on the left and higher resolution, we want to know what's the advantage of different dimensions and what are the ones that we can get a lot of data of, because if you have, say, someone with quadriplegia and you, for research purposes, put in these wide microarrays, you'll get tremendous information, but you're only going to have a handful of people who are candidates for that approach. Whereas if you take advantage of clinical opportunities, then you can quickly get much larger numbers. So the things that make a lot of press going back almost 30 years now have been the efforts by BrainGate, which came out of Brown and Harvard and several other areas, to take the Utah array, which came from Michigan Technology, to place these arrays of shanks into motor cortex, decode motor signal, and basically take a very what I'll call a phrenologic approach, that you look for a neuron that moves the hand and then give that signal to the robot to move the hand. And that works. And I don't have the video here, but this woman was able to move for the first time since her pontine stroke, a intent to emotion, and then was able to give herself a drink. And then on the bottom is essentially the same approach with modern technology and much, much better computing that comes out of Neuralink and the Elon Musk world. As you see, very similar dimensions and very similar approach some 30 plus years later. We observed, along with many others, that if you look at the corticography and open up the signal to a much higher sampling rate, so you have to sample at 2 to 4 times the signal that you're interested in. So once you can get up to 500 or a kilohertz of sampling, you can start to talk with good signal about these higher frequencies. And then if you look at the higher frequencies, you can see something that resembles our understanding of motor cortex, meaning that there's a patch of a couple centimeters that is active when you move your hand. And then if you look at the lower frequency, you can get movement dependent changes, but they're much broader spatially. And so you're able to do this and reconstruct the classic homunculus. You can see hand movement that's more superior medial to the more inferior lateral tongue movement. And especially in the high frequencies, this is very separable, even in an individual, even over just a few seconds of recording. So very, very powerful technique for traditional mapping. Now, what the University of Washington, among many things, is famous for is work in our national primate lab. And if you go back to 1970 and then the work just before this, comes out of a science paper. And these papers are really fun to read because they're very narrative in their text. It's not the staccato sectioning of methods and others that a modern paper would require. The information's all there and it reads very well, but it's a very different format. And the photos, of course, are not CGI. They're all snapshots from the oscilloscope or other tracings. And so these are, in the top is what you would think of as a traditional record a neuron, which is U, and then look at the EMGs from different muscles, which are the other letters of the alphabet. And that's flexors, extenders of the wrist, and biceps and triceps of the animal. And so you see when the motor neuron fires, you get motor activity, and that's great. What Dr. Fetz, who only recently retired after many decades of a seminal career, showed is that, first of all, it can change. So if you reinforce the animal appropriately, now the exact same neuron is firing in completely different pattern of muscles that form. And then most intriguingly, is that you can also dissociate it completely. So in the bottom, it's the exact same neuron, and you've simply, not simply because it takes a long time to condition the animals, but now that same neuron will fire and no movement occurs. So this means that there is the ability to volitionally modulate the interaction between even M1 and the output. So it is not a one-to-one, or at least not a static requirement. So what we see here, sorry for the extra text there, is what we did in our early work on brain-computer interface, which was a simple feedback task that took the power and the high frequency and said, okay, when you move your hand, or tongue in this case, we know the power goes up. Let's now link that power to the position of a cursor. And what we found over time is that not only could folks within a couple minutes figure that out and move the cursor, but they actually stopped thinking about the movement itself and instead thought about moving the cursor. So using the exact same electrode, they would take the power in that electrode and increase it beyond even what you saw for movement. So just simply by using it for this different task and giving that feedback, you can volitionally increase the power over motor cortex when you're not doing any movement at all, but actually using it for a completely different output task, one that evolutionarily or in that person's lifetime they've never done before. And we know that cortex cares a lot about what you do. So just even from a comparative anatomy point of view, how motor cortex is distributed and amplifies different parts of the brain. It depends a lot on your environmental salience. So we have a lot of cortex dedicated to hand and mouth on both sides. The rabbit cares about what the rabbit does and so forth. And even amongst the mole species, there's a difference in what the cortex cares about, whether it's your whiskers, teeth, or whatever it is the star-nosed mole is doing. And this can change over years. So if you have injury, and this is from amputee literature, then what you'll see is that, and this was recapitulated in animal work as well, that neighboring areas, especially the face, will migrate into these areas. Again, whether migration is the right term or unmasking of previous projections isn't clear. But what is clear is that these parts of the brain that are themselves not damaged are able to repurpose to retained projections, retained relevant tasks. And so we asked the question, well, in the setting of these brain-computer interfaces, if we have the homunculus, is it really static even, and can we modulate it even in this short period of time? And so what we did, among other tasks, was this, where and Luke Bashford, who had come from Oxford by way of Freiburg, Germany, helped with this task and asked the question of, if you've got a piece of brain that's very linked to motor cortex. So when you tap an arrow, left to right arrow, this is the part of the brain that's most active. And we said, okay, we're going to take that exact same piece of brain and drive the movement of a cursor up or down, depending on the activity of that brain. And we want to do that at the same time that you're tapping an arrow. So really, it should be impossible to drive the cursor down and to the left or right, because you're tapping the arrow, which should activate that piece of brain, which should move the cursor up. So anytime you're tapping an arrow, you should be moving the cursor in the direction of the arrow and upward, and never be able to move it downward other than the complete middle, which is not doing anything. And we tried it, same cortical area driving both components. And what you find is this, so if you look at either these two on the left or these two on the right, it's the exact same hand task. You're tapping an arrow, but in one case, you're driving the cursor up, which you did by this band here between the two horizontal lines, which is red, which means it went up. And here you did the exact same overt motor task, but you're driving the cursor down. So you are able to, in real time, dissociate the function of this piece of brain that we picked specifically because it was tuned to hand function. And so that we can take the homunculus and modify it in just over the course of minutes. So with that level of flexibility, we in 2010, Yoki Matsuoka, who is a robotics researcher that came from Carnegie Mellon, and then went on to work with the smart thermostat after a few years with us. But she and then Rajesh Rao and Tom Daniel and then Chet Moritz later ran what was called the Center for Sensory-Motor Neural Engineering. And this was a really neat funding mechanism from National Science Foundation that's 10 years. Funds are matched by the university, funds primarily students that are in collaborative projects. And so from this evolved this concept of using the innate plasticity of the nervous system to drive a variety of outputs. And that output could be, at that time, robotics was kind of the hot topic, nerve and spine stimulation, some of the work that Chet and Christoph in our department have been working on, and others. The neurochip, which is this idea of doing processing close to the brain and then stimulating other parts of the brain in response, but it didn't really matter. The bottom line was the philosophical concept that you're going to tap into the brain's own recording. And in this template over the past 10, and with that 10-year grant, we looked at the different components at the bottom, which included an explicit look at neuroethics, computational neuroscience, looked at whether you needed different electrodes, different computing. And then at the top were the systems. And at this point, now 15 years later, with this study, we have now checked off all 3 boxes because Chet Moritz and Onward, which was a spinal cord stimulation company, looked at reanimation. We have the study that Jeff's going to talk about with that was done with him and Medtronic and Andrew Coe, looking at volitional control of thalamic stimulation, and then with this study, explicit use of cortical plasticity. So let me turn it over to Jeff and talk about the first of these studies that was done now over 10 years ago in collaboration and using the Medtronic device, and was the first example of using intent to drive stimulation in people.
Yeah, thanks, Jeff. And one quick comment. There seems to be a Microsoft Auto Update has chosen now to put something up on your shared screen. I didn't know if you wanted to try to move that or not. All right, so given all this work that was going on at the University of Washington, looking at motor imagery and movement intention to basically drive cursors on screens, at some level there was this outstanding question of how can we use motor imagery and basically motor sensing of the motor cortex for therapeutic uses? And one big area that was basically identified through conversations with Tim Dennison at Medtronic, with Howard Chiswick, my PhD advisor, myself, and Jeff, was that we could use movement imagery as a way of titrating when and how someone's using their limb, so that we could basically use that for treatment purposes. Specifically, essential tremor is a condition treated by deep brain stimulation where people only have uncontrollable tremors when they're volitionally moving their limb. So we have a control signal now. So we have a control signal with this motor activity that we could use and sense from an implanted electronic device to then investigate how we could modulate stimulation in real time to maybe better enhance treatment. The device that we were working with was the Medtronic Activa PC+S, which was an investigational use. So this wasn't a commercial device. It required us to go out and get an FDA investigational device exemption on top of the normal IRB approvals. Andrew was the surgeon who implanted the device, and the additional piece of hardware that we had in these cases was a spinal cord stimulation strip, which was actually inserted through the same burr hole for the depth electrode to the thalamus, but then slid backwards such that it would rest along the center sulcus, giving us a couple electrodes on the sensory cortex and then a couple electrodes on the motor cortex. And we could then use this system by integrating in with a bunch of, honestly, a lot of software programming to basically do real-time analysis of the data, and then we used wearables to then look for outcome metrics in terms of tremor power. Jeff, would you hit the next slide, please? Why is it back? So actually, just go ahead. Would you just go ahead and hit? Yeah. So using this device, we then first thing that we did when we had our first implanted patient was we looked to see, can we replicate prior results with the that we had already previously seen in all these ECOG studies. And the good news is that yes, we could not only see large scale power band decreases on the left with overt movement on basically between rest and arm, but then we could also, in both the stim on and the stim off conditions, but then, in kind of a time axis ways, these desynchronizations were highly time locked and easily to sense, as seen on the right, where we have with rest periods, we have spectral broadband increase in the beta power. Now, one thing, of course, that we weren't able to see with this device was the gamma activity, and that was mostly because the system was fairly quiet. But this demonstrated that we were able to develop a classifier to detect on versus basic movement or rest states. Let's go to the next one, Jeff. So in 2017, this is one of the papers that I actually got out after I had already left, but we had completed completed the development of a fully closed loop system to work with these patients, where effectively we were in real time streaming neural data from this implanted device, performing some signal processing, and then turning the device, and then turning the stimulation on or off based purely on this motor signal of intention that we had decoded in real time. And the results of this study were quite promising. Jeff, next slide. This is one of the tasks that we performed, which was a repeated movement to the nose. So, and then we are averaging all the movements across the trials into these similar plots. And you'll see that if node stimulation on the very top plot, we have a lot of activity, and that's because that's during the activity periods where they're trying to touch their nose and they're having a lot of tremor, right? So that's what tremor looks like, is in the top left. Then we have the rest periods, of course, where there's no tremor, and that's we have these kind of large deviations kind of at 0 and near 10, which are the actual movement from the rest position to the hold position. You'll see that during open loop stimulation, there's no activity between the big deviations at the start, kind of in that 3 to 10 second time frame. That's because his tremor is well treated by stimulation. Notably, in the closed loop stimulation system, you'll notice that there was some small amount of tremor at the very start, but it was rapidly treated, and that's there was latency associated with the system at some level. There was some technical pieces that resulted in latency working with this particular device, but ultimately, from the patient's perspective, they reported that as long as it was predictable that tremor would go away, they were actually pretty satisfied with it. And so this basically really was the first closed loop demonstration of a BCI, brain computer interface technology, in a therapeutic application here, using this motor intention for something other than just driving a robot arm, as Jeff was introducing earlier today. On the next slide, we have a brief video which should play. Just kind of showing this in action, and you can kind of see 3 different spirals. And you can notice in the closed loop system, it's really indistinguishable from purely on DBS. Now, why does this matter when we're basically having a similar therapeutic outcome? Well, extra stimulation is associated with potentially side effects for some patients, but also at some point, a lot of these devices that are being implanted aren't necessarily rechargeable. So at some level, the more you can save power by turning the device on when it's not needed, the longer the battery will last before it's needed to be surgically replaced. So in terms of both power and possible side effect kind of incidents with these patients, these closed loop therapies are really a big kind of a step forward. And in other applications such as Parkinson's disease, the adaptive technologies are actually being shown to be better than state-of-the-art open loop therapy in terms of outcomes as well. Back to you, Jeff.
There we go. Great. Yeah, thank you. So the other, sorry, this will keep popping up. There we go. So the other 1969 to 70 finding that evolved in the lab was this idea of using the activity of one area to drive stimulation of another area. And so this was a way of reconstituting very classic Hebbian plasticity. So 2 neurons fire together, then they will develop connections, or at least enhance existing connections together. And so the idea here was that you're recording from one neuron, and then a distant one is stimulated within about 20 milliseconds of the activity of the first, and then you will actually change the output of that recorded neuron. So not the stimulated one as much as the one that's being recorded, because the other area is now activating a constellation of muscles, and then that drives the map of the first. So it's a little bit of a not straightforward concept, but it means that in this case, so this is in the space of muscles of the animal, radial and ulnar activity and flexion extension of the hand, and you have a neuron that's very interested in this flexion radial rotation direction, quite well tuned. But because you're stimulating at the same time that it's active, it changes its tuning in the direction of the other neuron. that's remote and being stimulated at the same time. So it's that coincident stimulation and activity that engages this same neuron and alters its profile over just the course again of a few sessions. So we looked at this and said, well, what are the opportunities to do this? And we do know from many other fields, especially up in the New York area, that we can see very robust cortical potentials in our intracranial patients. And these are signs of connectivity. And so we asked the question, can you change these measures of connectivity using this method of activity-dependent stimulation? So then Stavros Zanos, who had done a lot of work with our department and was a PhD student in the FETS lab, went back to the primate model, had electrodes that looked more like clinical electrodes, so these subdural ones, and did the same experiment where it would stimulate at a medial frontal site during the activity of a motor area, and found that especially if you stimulate during a very specific part of the cycle of the activity, that you can get this same enhancement of connection. So again, if you look at where the red tracings are, if they're in the trough of this cyclical behavior, this is again our friend the beta rhythm. If you stimulate in that trough, which is a time where the cells and population seem to be more active and more open to this connectivity, then you can get a very strong up to doubling of the strength of the connection as measured by CCPs. But if you're in the hyperpolarized state, then you don't get that effect. In fact, you can get an inhibition in the animals. And you see the data is distributed certainly, but you can see the pattern that if you're in this depolarized half, you can get an enhancement, and you don't see it in the hyperpolarized state, if anything, a diminution of that activity. So then we went back, and this is in itself a bit of a story because we could only find at the time, this was 2008, and we had written in conjunction with a collaborator in Eastern Washington, a life science discovery grant. This was back in the days of the tobacco money settlement, and got money to do this. We could only find one stimulator that would accept an external trigger at that time that was FDA approved, and this was made by Cadwell. And it was actually a stimulator that was being sent to be mothballed at Children's, and we had put a note that we were going to use it. And I was walking by the loading dock one day, and with a pile of things was just about to be picked up, and there was the stimulator with a note that said, "Do not dispose till clearing with Dr. Ojemann." And I said, "Okay, well, I guess this is the moment," and grabbed it, and then we did the study. So what this experiment was trying to replicate what we had in the primates, which was that you have one electrode that you're stimulating. It has connections to both the control and to the recording area. But if we stimulate at the time of beta depolarization at the recording site, then we will enhance the connectivity between those two areas. So that was the hypothesis. We went ahead and did the recording. We had both hyperpolarized stimulation delivered on the hyperpolarizing phase at the negative crossing, and then one with a playback, which had no phase dependence, meaning we just took some random segment of data and stimulated at random times to see if there was any effect of stimulation independent of phase. And what we found was that with this phase triggered stimulation, you had a dose-dependent effect. So if you had one stimulation in a row, you got a certain amount of increase in the strength of the peak, and this could up to 5 in a row could greatly strengthen up to about 5% the strength of the CCP. And we were actually able to do this in a patient who had a neonatal stroke and epilepsy and enough preserved hand function that warranted intracranial monitoring. And so we were fascinated to find two things with him. First was that we did find relatively preserved beta and high frequency gamma changes when he moved his residual hand function. And that second, we could induce these same changes in the cortical evoked response. So this was in an N of 1 at least evidence that what we were finding in motor healthy patients could be extended into people who'd had previous damage to cortex. And we saw some evidence of phase dependency. This is not a graph I'm the most proud of. We do think with some of our newer devices, we can get a much more precise delivery of phase, and evidence is that will show even more precise and mimic what we saw in the animals in terms of the selectivity of the phase. So we took all that together and took another finding with Seattle Roots. And this was a trial that had been done by North Star Neuroscience that involved many names that are connected to this department previously. Alan Weiler, former resident and faculty, was involved in the implant of these. Steve Kramer, who's now a PI on our study, was a neurologist, along with Claire Kreutzfeld, who's joined us this morning, and longtime university resource. Well, we're not resources, right? We're stars. And what they found was that stimulation of the brain around a prior stroke during therapy would induce recovery. And this had a very successful phase 1 and phase 2 trial with a new device. And then when they went to their pivotal trial, which was a large randomized trial, they had a lot of patients who had no intact corticospinal tract. And so they wound up not meeting their primary goal. But when they post hoc broke down their goal, which is what the bottom panel shows, is that if you took the patients that had residual evoked response, so if you could stimulate any part of motor cortex and see a response, albeit impaired, that group did exceedingly well and maintained their benefit past the stimulation and would have met criteria. But there were enough in there that did not have any preserved response that contaminated the trial. So with that, the company was unable to do the next round. Venture capital funding disappeared, company went bankrupt. And there we were for about 10 years with not much going on in the stimulation for stroke world. And Jeff, do you want to talk, maybe because you know a lot of these as well, where did that put us in the past 5 or so years in closed loop applications?
Yeah, in terms of closed loop applications, really with both the Activa PC plus S, but then with the successor system, the Summit RC plus S, which was also developed by Medtronic. And that was what I spent my time at Medtronic working on, was the successor system, the original Activa PC plus S. There's been now closed loop research using these implantable streaming and stimulation devices in OCD, depression, chronic pain, but of course also the traditional movement disorders treated by deep brain stimulation, Parkinson's, and tremor, of course. And then, of course, there's this exciting new study that we have some more recent results working now in using all these, leveraging the combination of closed loop therapies along with all this neuroplasticity work that Jeff just reviewed, in terms of now trying to go back and taking a neuroplastic approach to that Everest study in our new stroke work that is going on here, where we are explicitly trying to induce plasticity. One notable thing is in many of the deep brain stimulation work for even in the adaptive sense for Parkinson's and tremor, you turn the stimulation off and the symptoms are back exactly the way they were before. So there's improvements to therapy, but really at some point it's hard to kind of suggest that we're really inducing any sort of long-term neuroplasticity for any sort of effect beyond just basically doing treating symptoms. But in this next generation of work that we've been working on, we're talking about now devices that are potentially being used to rehabilitate stroke patients, where the device has almost like a limited time where it's needed, and then hopefully by inducing the neuroplasticity, patients will not need the device anymore. Jeff, let's go to the next device. Next slide. And so in these, this work in towards pulling together a system for a clinical study, such as what we're doing in stroke, really, there's a lot of questions that we need to start to ask and answer in terms of, what are we using as the biomarkers for for enhancement? How does this differ across patients? How much sort of resolution do we need? Both in terms of resolution in terms of time, but also in terms of space with the electrodes, and then also with what sort of resolution in terms of bit depth or fidelity that we need to pull it together. And then, at some point, we also need to start pulling together questions of how do we explore the potential stimulation application space in these work. And this longstanding historical work in inducing neuroplasticity, both out of the FETS lab and out of the work that's been going on here in the grid lab in ECOG, it really provides us a lot of places to start with that. So let's go to the next slide. So we constructed our study around using the Cortec brain interchange device, which is a collaborating manufacturer of devices out in Germany, in Freiburg, which is in kind of southwest Germany, close to France and Switzerland, right up against the Black Forest. The device has 32 sensing electrodes that we can work with. So you'll notice in this top left picture, they're a little bit exaggerating what they can do in that one. But the device has 32 electrodes that we can use for sense and stimulation across any of them. The device itself actually isn't powered. There's an external unit which then delivers power to the implanted device from a laptop. The laptop is actually, the device also does no processing of the data. So in order to make this system work, you have to program and pull together a system to basically run the device in real time. And in our study, the device it's a head-mounted device, and in our study, we were going to use the electrodes as a subdural implant with the electrodes directly on top of the brain, spanning once again this central sulcus with both sensory and motor cover and premotor coverage. Next slide. So, at some level, there's a lot of work that goes into even before we start having to work with patients. As I mentioned, there's a lot of system programming the systems from scratch is something that is neither fast nor necessarily easy. This is one of the areas where we've teamed up with collaborators out of Oxford, Tim Dennison's group, on top of working with Cortec and my PhD student Hanbin, focused on developing kind of a framework for us piloting and testing these algorithms that we want to run in the patients in kind of a responsive saline tank that allows us to do things like look for assess how does the device handle simulation artifacts. Are we able to do the phase specific plasticity, but also then take a look at things such as, can we simulate an evoked response using an active circuit that senses when the device under test, the brain interchange, stimulates and then renders a real-time response, so that we can then basically develop the data analysis pipelines that we need to run our study. So given this work in establishing feasibility, we also actually then took this device into the epilepsy monitoring unit for kind of final functional tests. And we were able to kind of show that, we were able to maintain phase specificity, and the system was able to work as designed. Now, doing the system integration, while this is still wrong, I can talk about this for hours. I'll spare you. This is only the first step, really, in the prep towards the implant. And really where a lot of the work goes into is less on the engineering and more on, frankly, the regulatory side. So Jeff, let's go to the next slide. And while we just had a recent first case, I'd like to emphasize that this journey really kind of began in 2019, when I was first joined the department. We submitted several rounds of investigational device exemption pre-subs to the FDA, focusing on the potential use of Summit RC+S. Before, in September 2020, we had pivoted to using the brain interchange device. And so at some level, we were able to make the argument that a lot of our previous conversations around the protocol and the inclusion criteria for the Summit RC+S should carry over into our new discussions around the new grant. At some level, there was a lot of the brain interchange was an untested.
Sorry, Jeff, do you want to just explain to everybody, because this was news to me, like what is actually a pre-sub meeting and what is the process?
Yeah, so there's different types of meetings you can make with the FDA. There are formal investigational device exemption application that you can submit, but a lot of times you just need to basically have a conversation and honestly work with the FDA to design something that would pass their review. And so a lot of these early discussions with the FDA were talking about it was a discussion around, are we recruiting patients who are 3 months post-stroke or 1 year post-stroke? and negotiating in a frank discussion, basically identifying that it seems like 6 months is something that is acceptable for both parties. So this is where the FDA traditionally has had a lot of willingness to work with sites to basically try to identify to basically work through the thinking process of both sides and through the how do we justify the different decisions that we're making in the design of a study that we'll pull together. Now, notably, the FDA will not actually look at a device's file until there's basically a submission, a full submission for it. So part of the way that the whole like the ID process is kind of designed for a site like us who isn't necessarily who's making use of someone else's device is that company pulls in a master file of all of their design history and all their secret IP, basically, more or less, that they can make available for the FDA to review for the device. Which then basically we can focus our IDE submission not on the functionality or the biocompatibility testing of the device, but just in the use of that device in our specific protocol. But even still, through a lot of these Q subs and these first IDE submissions, there's a lot of back and forth with the company as well, right? So in many times, a lot of the things that are coming back here are also potentially indicating to the company what they need to make sure is in their master file, because we can only submit our full IDE submission when the quality management documents are completed by the company. And that's actually what largely drives the timeline in this case. Towards our first full IDE submission in 2022, the company, CoreTech, honestly had to keep on working and pulling together the final components. So instead of waiting until this company finally has everything pulled together to then submit an IDE, the pre-sub process also allows us to get the ball rolling early on working through all the things that are protocol specific. Now, in that full IDE, there was a lot of submission. There was a lot of discussion around the safety of the device. We used NeuroPace as a example system, which is also cranial mounted that needs some milled surface. They also requested a lot more information on all the steps we do for the inclusion criteria screening, such as more details around the TMS that we were using. Also, a lot of pushback on what score ranges were acceptable. So even though we had all these back and forths in these pre-subs, at some level, it's not until the full IDE submission is made that they actually look at the potential risks, benefits, and the trade-offs here with the actual safety information about the device in order for them to make to review perhaps some of the things that weren't necessarily the focuses of discussion, or even the things that were focus discussions. Pre-subs aren't binding. Full IDE submissions are binding, though. Now, there was some things that actually came up in this whole process, because in 2022, the FDA had actually, through COVID, had lost a lot of their senior reviewers, and there was frankly some junior reviewers. And actually that's on the next one. But sorry, actually I was a little premature there. But they did ask for a big full cybersecurity risk assessment. So in modern day, cybersecurity is a big piece of how any of this works. It's really critical that everything basically go through a cybersecurity assessment. But honestly, the biggest piece that came out of this was that they indicated that we needed to just focus on safety evaluation. We shouldn't be focusing on neuroplasticity. And now, Jeff, we can go to the next slide. Apologies. And basically, so we had to completely rescope out our protocol to basically we had started out with our initial submissions with a protocol that had 2 groups and a crossover. It was a blinded crossover study. We had to get rid of all of that because the FDA wasn't confident enough yet in the safety of the device. They wanted to basically or necessarily the applications of some of these neuroplasticity inducing paradigms. And they said, listen, you're coming at us with a new device and a new protocol. We're comfortable with one. And because it's a new device and you're stuck with it, you need to focus on safety and feasibility first. So we redesigned our protocol now, where we're using the open loop Everest style stimulation, i.e. the old protocol from Everest. where we're doing tonic stimulation. Now, the EVER study, as Jeff reviewed, showed that if connectivity is intact, and we're recruiting patients whose connectivity is intact, this should provide benefit. And then we are going to be doing a long-term monitoring period where we're going to be basically evaluating how the device is functioning in a stroke patient. And then we'll have the opportunity right before we explant the device to basically try out and collect some preliminary data with these more neuroplasticity inducing measures to just basically evaluate if they are feasible to use with the Cortec in an implanted stroke patient. And so this was the new study that we have proceeded with. And so then on the next slide, there was and then of course we go through all this design. There was another round of IDE, and this is where they actually identified that they wanted Cortec to completely recollect all their animal biocompatibility and histology data. And so that's a more than 1 year delay. And this was because at some point across the field, this wasn't just Cortec. FDA in 2022, 2023 raised the bar of what was required, and basically every single company who makes devices who is going through a regulatory submission had to more or less recollect their animal data. This wasn't a Cortex specific thing. I think even Medtronic had to go off and recollect a bunch of animal data. So it wasn't until April 2024 that we finally got IDE approval. And this is where then the IRB, of course, reviews after that approval. So April 2024, we submitted our final version of the IDE. Jeff, next slide. We finally got an approval. By this point, we had 3 full submissions of the FDA IDE. We had 5 pre-subs, and we had at this point a 16-month no-cost extension. And then, we were celebrating at this point, but we didn't know that we would then take 6 months to get through the IRB for review. So we were finally fully approved in November of 2024, of course, right in time to try to start recruiting patients in the holiday season, which is not a good time to start recruiting patients. Of course, then in January, we had another complication. Not necessarily in January, but in January, there was some complications started percolating through the process. Notably, there was a new release of information, of a policy shift where foreign collaborators could no longer be a part of grants. And while we didn't necessarily, we were working with this German company, they weren't necessarily there was no subaward. So which is why we initially squeaked through this, the speed at which this policy update was kind of released meant there was a lot of confusion and kind of difficulty in terms of how we go about whether or not there was a period of time where I think our study was on hold in part because of this, and finally it was resolved after several more months of basically no word from the NIH, but we did get the go-ahead eventually. All of this to say is that 2 weeks ago, when on the next slide, when Andrew and Ben basically had the first case putting this device in, it was quite the milestone for all of us working on this team. It was a real pleasure to be in that operating room working on it with them. And on the next slide, Jeff, we have some exciting images taken of post-operative CT down below shows you just what this surgery looks like, with both a craniotomy for the electrode placement and then also a milled recess for the cortex brain interchange. We have the locations of the electrodes laid on top of stroke, basically the areas impacted by stroke. And it was quite an exciting time to be in there.
Yeah, this is just so everyone knows, this is Kurt Weaver put this together. He's joined in our faculty with radiology, and you see the preserved functional MRI response, which was also something we validated to make sure that there was preserved areas. You see his corticospinal tract and how it's disrupted, smaller than on the unaffected side, but still present, and then the stroke itself based on the T1 signal change. So putting it all together gives us an idea of a target of areas that are still preserved, and can we recruit additional areas into the process of recovery. I just got a text from Dr. Ko that he is doing very well. They're very, very anxious for this, for the patient. They're very anxious for the process to start. So this is probably the toughest slide to put together because there are so many people over 50 years. Several of our collaborators who are on. I saw Drs. Kreutzfeld, Weaver. I'm sure I missed a few others, so apologies. Steve Kramer was here now at UCLA. Was here about 20 years ago, and is the co-PI, and he's been doing a lot in various stroke recovery trials. The center that's now the Center for Neurotechnology and sits in one of the Allen Engineering buildings. We've had residents. I was trying to think of this morning. It's David Sue had worked on this project. Our own Andrew Ko, Kelly Collins, and our own Rob Buckley have all been involved in various parts with this. And then I think we never forget the courage and necessity of people willing to put themselves through these things, always in hope of a better future. So I hope this inspires, it certainly does inspire me and inspires everyone to continue our progress. This is what we talk about when we make the case for why society should invest in science. This was if you add up all the various funding that goes into this, is probably well over 100 million over those years that has been put into this. But it both pays for itself, not just in sheer dollars, but in human impact, if in fact we can whether this product or other products get over the finish line. Thanks for your attention.
It's a standing ovation by all of us. Thank you for staying with this project. Fascinating. I'm not gonna. People are clapping. I see. Any questions? Questions? Fabulous work.
Well, I have a question for Jeff.
I have
If there's no other questions, but
I have one question.
I'll go. Yeah.
Jeff, besides motor rehabilitation, what other things? What about vision? How about language? Are there are trials doing that too?
So I'll answer part and then turn to Jeff H. If you go back to the original, sorry, the original North Star trial, they very quickly went to different applications. They did auditory cortex to suppress tinnitus, but they also did stimulation of dorsolateral frontal cortex for depression, and then stimulation for aphasia. This should work for aphasia, this idea of recruiting additional areas. The other thing that's kind of exciting is if you look at the aphasia literature, contralateral recruitment may be unhelpful. And so different stimulation patterns could also suppress maladaptive plasticity at the same time. So these are all things that are out there. I'll let Jeff talk about some of the depression literature, but this approach is distinct from what others are doing on with visual cortex prostheses, which is really going back to that one-to-one. If you want to activate this phosphine, stimulate this part of the brain. And so when you think about sensory cortex prosthetics, you might similarly do that. But I think even in those areas, the ability of the brain to interpret input in a dynamic fashion when coupled with salient activity. So one of the other key pieces of this is all this is done while you're doing physical therapy. So the message to the brain is throughout this is that all this information that's coming in, all the stimulation and noise is relevant to the task at hand. And that also seems to be key to enhancing recovery. So Jeff, I don't know if you want to comment on some of the other applications that we mentioned in that slide briefly.
Yeah, I think at some level, taking this neuroplastic approach, really it's emphasizing the connected nature of all the brain, right? It's not just a stimulation target anymore, but at some point, these are brain circuits that basically, by influencing how information is flowing through these circuits, perhaps we can change behavior. I think that in large part, we know neuropsychiatric space is largely dominated by maladaptive circuit that need to be adjusted in different ways, right? So I think the neuropsychiatric space is particularly ripe. Now, at some point, I think the neuropsychiatric space, just due to the amount of time that's been worked on it, I'm not going to call it behind, but it's just it's taking a lot longer to really kind of start identifying neural markers or that we could potentially use for like outcome metrics as a surrogate for a behavioral assessment, right? In many ways, devices that are working in closed loop ways to maybe perhaps monitor and look at it, something, you know, a connective space of how something's working. If you relying on a psychiatrist to do an assessment to basically understand how the system works, makes it very difficult sometimes to try to automate the methods to basically try to, like, you know, use more advanced machine learning or AI
Yeah.
to pull it apart. So at some level, there's a lot of opportunities there. And I think actually that, you know, some of the more typical connectivity measures, evoked potentials or resting state coherence, might be actually good midterm investigation opportunities to look at.
Yeah, I think the beauty of what you have is you have a target. You have motor function. Okay, psychiatric targets are too disparate and
Yeah.
too complex, but you pick motor function in something that could be measured and tracked. That's great.
That's the place to start, and then we'll do the more complicated stuff later.
Yep. Thank you both. Fabulous. Anybody last quick question or just thank you everybody for listening to this. This is really phenomenal, cutting edge work.